All right, guys, any questions before we get going with this stuff? All right, so we've got about half of chapter 4 left. There are a few details we will go into, and then hopefully I'll be able to start chapter 5, maybe do it all. I don't know, I doubt that. Chapter 5 is relatively short. There are a handful of things in there, but I also feel like, well, in both, it's kind of got some extra detail that don't really need to get super bogged down with. All right. So for those of you tuning in, if you have questions, unmute yourselves and let me know, because I usually don't, if the chat pops up, I typically don't see that until much later. Okay, so we left off, we talked about how enzymes function. The littlest bit, we talked about it. We're going to go into some more details and show you a little bit about some of these ideas, all right? So with that, okay, so with that, we showed, we did go over this slide last time, but ultimately the idea, the red dash line is talking about what's happening with an enzyme. So this is a chemical reaction. There are all sorts of different types of chemical reactions, building reactions or breaking down reactions. So anabolic or catabolic, we've mentioned, introduced them the littlest bit, all right? In this case, we're going from lower to higher energy, so we are building some bigger molecule, a macromolecule. It's an anabolic reaction, all right? And it takes energy. We said those reactions aren't spontaneous. Talked about that a little bit, all right? So with that said, in order to make some of these reactions happen quicker or to happen at all, oftentimes it's not going to happen without an enzyme. It helps to reduce this activation energy, that little hump that you've got to get over in order for those molecules to do something, all right? And it doesn't matter if we're talking building or breaking stuff, it takes energy to get these molecules ready to do that, all right? In a catabolic reaction, when we're breaking things down, it doesn't take a huge amount of energy, but you still need a little bit, all right? So enzymes, doesn't really matter which way we're talking about, enzymes help to speed up these reactions. We say they're catalysts. They help to make it function. And like I said, just a second ago, many of these reactions aren't going to happen at all unless the enzyme is present, all right? Especially thinking building reactions, all right? There are courses, biochemistry courses, that is really kind of the main focus, especially like in the lab portion of it, is thinking about how these molecules interact and calculating rates of reactions, how quickly these things happen, and typically thinking like seconds for it to occur, right? So enzymes are types of proteins, and there is all sorts of differences with these enzymes, right? They have specificity, so they act a lot like a lock and key, where that lock only allows this specific key in them, right? And if that key goes in, you're in business, right? I like to use this example. I saw this happen many years back. I was at the grocery store. I was leaving. I had my cart full of stuff, and I witnessed this lady, little old lady, freaking out, right? As I am going toward my car, my first thought is somebody is trying to steal her car, all right? Which I think is what she thought as well, all right? So there is this guy at her car trying to put a key into the door, and she is freaking out. Again, this is her stinking car, right? As it turns out, this poor gentleman was not trying to steal her car. He had the exact same car the next lane over. All right, there it was. And he's like, what? And he looks and he's like, oh my gosh, I'm so, whatever, there's my car. Same color, same stinking car. The key fits in, but it didn't turn the lock, right? Enzyme substrate, right? It looks the same, but it's not, right? It's got to function exactly, all right? So it depends on the types of reactions for the specific types of enzymes. Some enzymes push things together to build stuff. Other enzymes put strain on bigger molecules to break them apart. Just depends on the type of enzyme, but that's really kind of what they're doing as they're allowing through these interactions. So this is a catabolic reaction where we come in with a substance, this green guy, a molecule with two different things linked together, and we put some strain on it, splitting it into its respective bits, the monomers, all right, the pieces. And in doing so, in a catabolic reaction, when we're breaking these bonds, we release energy, right? And it's that energy that we want to think about, all right? In a chemical reaction, can we harness this energy? Could we use that and convert it into energy for our cells, recharging ATP molecules? That's really what we want to think about, right? There are boatloads of different enzymes and boatloads of chemical reactions, right? And we talked about metabolism as both the building and the breakdown, right? And at the end of this chunk of stuff, at the end of chapter 5, I stole a picture of biochemical pathways that are in a big chemical book where you can order all sorts of things. And it's got these pathways spelled out. We are showing you stuff, and I'm pretty stinking sure as I move through and you see some of these reactions, you are not going to say that's simple, all right? And I'm going to try to simplify it even more for you, all right? But I wanted to show you at the tail end the true complexity that we are really glossing over, all right? Because there is all sorts of business going on here, right? And these are like reactions that sometimes can be going in the opposite direction. So just because we're breaking something down doesn't mean that we're not building the same crap later on down the line, all right? It truly depends on these circumstances. different enzymes participating in it, but we want to think about this as a whole, all right? There's a lot of stuff happening, all right? And we definitely try to, kind of simplify it, all right? There are different ideas with this as far as how some enzymes might potentially function. You don't need to get bogged down with this, but we may inhibit an enzyme or we may activate an enzyme. And in these instances, we call this allosteric because it's a different area, all right? It's not this active site, all right? That's the business end of that enzyme. Let me back it up. The active site right here is where those substrates, the green guys, come in and link up, all right? So the active site is available, the substrates come in and we do something. We can inhibit or activate these reactions allosterically by stimulating this enzyme at a different region, right? So inhibiting it, some other molecule links up to this enzyme and it makes that active site change shapes. So instead of being open for business, now it's closed, right? Or the opposite, with the activation, we link up to that enzyme, which is closed for business. And once the activator links, we open for business and the active site's available. So it depends on the types of reactions, the types of products and substrates that we're dealing with that allow for regulation of this. Some stuff gets regulated and other stuff just happens, all right? It depends, all right? They're not all governed by allosteric mechanisms, right? There are some other types of mechanisms as well. And let me just back it up to this real quick. A competitive inhibition would be where you've got a different colored substrate so it can fit in there, but it doesn't act on it. It's like the guy trying to open up the wrong car, right? The key fits, but it doesn't turn, right? So as this is happening, he would be the inhibitor. He's in there with his key in the lock and the little old lady can't get to her car because he's blocking it. His key's already in there. So he's inhibited that. She can't actually get home, right? That would be competitive inhibition because he's put the key in the lock, all right, to the active site. So we'll skip that. Oftentimes, with some of those inhibition processes, we think about some end product serving as the inhibitor, and this would be allosteric inhibition. We're not acting on the active site, all right? So numerous reactions are going to be a stepwise approach, where it's not just one enzyme or one substrate to a product, it's multiple molecules. And you start with one thing, maybe it's a big macromolecule, and we will go through numerous enzymatic reactions where the product of one initial reaction becomes the substrate of the next, and we just keep handing them on. So here we can see three different enzymes, three different enzymatic reactions, the triangle to the rectangle to the crazy shape looking guy, whatever it is, all right? So as we move through there, substrate to product over and over, all right? So here we can see intermediary substrate, second intermediary substrate, and then finally, as this guy's acted upon, it's the final product. This is what we wanted to build in the first place. It just took three enzymes to get there, all right? And when we think about trying to harness energy or manipulate molecules, enzymes help to lower this. So doing it in stepwise approaches helps to maximize the amount of energy that we can harness from this system or minimize the amount of energy that we're putting into it to get that final end product, all right? That's the idea, all right? When we think about the total amount of energy, could you go from the triangle to the square in one shot? Maybe, but at a cellular level, it would be very energetic. It would be too costly for that to happen. Or breaking down a big macromolecule like a glucose that's taken into the cell and immediately combusting it into the carbon dioxides and water molecules. If you did that in one shot, that cell would explode with the amount of energy that's being released. So we do it in small little steps, all right? That's the idea, all right? So respiration and fermentation are two ways that we take molecules and catabolize them or break them apart, right? And with that, it is aerobic when we talk cellular respiration. And when we talk about fermentation pathways, this is anaerobic. So no oxygen is involved in these mechanisms, all right? So this is a chemical formula in the presence of oxygen. So aerobic respiration. In the presence of oxygen, we see this happening, all right? So this guy right here is a sugar molecule, C6H12O6. It's a glucose, all right? We need some oxygens, right, in order to combust this molecule. And the arrow is showing that this is a chemical reaction, and the arrow points to the products, right? There are some reactions where the arrows will be going both ways, and we will say that's a reversible reaction, right? We don't need to worry about that right now. Probably not at all, really. All right, so the arrow points to the products. So we start with the sugars and some air, and we create water molecules and carbon dioxide, all right? A quick and dirty chemistry lesson for you guys. When you look at the reactants, and each of the letters is a respective atom, the numbers are the amounts of those atoms we're dealing with, all right? You can add them all up, all right? And then you look at your product side, and they better be the same, right? If you don't have the same, it's not the same molecules, they're manipulated, but the same atoms, they're there. And if you calculate it up and those numbers don't match, somewhere in this process, you've made a mistake, right? Something's not right. It's a balanced equation, right? And in chemistry, that's kind of a check so that you look at both sides and you're like, the number's equal, I've done it right, all right? Hopefully you've done it right, all right? There still could be other issues, all right? But we've got 6 carbons here, there's 6 carbons over here, there's 12 hydrogens in this bit, there's 12 hydrogens right here, and here we've got, what, 18 total oxygens, and we've got 6 here, and we've got 12 there, 18 total, right? It's balanced. It's the same amount of stuff. We just manipulated these molecules around, all right? So one sugar molecule, one glucose, gives us 6 carbon dioxides, right? We need the oxygens, and we manipulate these other molecules, and that's where we get 6 waters out of the mix, all right? If oxygen is not present, then cells will ferment. And there are lots of cells that have the capability of fermenting. Truly depends on the circumstances, and the products that they create are also going to be different, all right? So we want to take sugars and we want to break them down, but oxygen's not there, so we will use a different process and manipulate these molecules, breaking down the glucoses and then going about it a different way, all right? So we think about stuff like in muscle cells, lactic acid being generated in the process of fermentation. If you are exercising and you kind of starve your muscles of oxygen, you wake up, or maybe even later in the day, you find your muscles are really pretty sore. And it takes a little bit of time to metabolize that lactic acid before that goes away, all right? If you're something else, you might go about it a little bit differently. There are bacteria that ferment and produce lactic acid that make yogurt, all right? Similar sort of an idea. Others, like yeast cells that can ferment, and they produce alcohols. They make ethanol in the process of this fermentation, right? Kind of just depends. And there are some other products that can be generated through fermentation pathways as well, all right? Not just lactic acids and alcohol. But those are 2 of the good ones. So when we think about these reactions, all right, whether it be aerobic respiration or fermentation, the very first thing that happens as we're taking glucose into the cell, the first thing we do is glycolysis. And glycolysis is the splitting of sugar, all right? Glyco is sugar and lysis is to split, all right? So the splitting of sugar. And this glycolysis, we'll see here in just a second, it doesn't matter what you are. If you're aerobic or anaerobic, every single thing that's alive on this planet goes through glycolysis, all right? What you do after that is going to depend, all right? But everything can do glycolysis, all right? So we're going to look at this and follow the sugar molecules as we move through, all right? And with that, there are a couple of reactions that we break into different bits, all right? So we showed you the sugar, we showed you the water and carbon dioxide that you get when you're done. That's just the quick chemical reaction. There are countless enzymatic steps that go from sugar to those CO2s at the tail end, all right? 3 different big steps with numerous enzymes in each of those steps, all right? So we'll see that, all right? The idea truly is to generate ATP molecules, the energy currency, if you will, of your cells, the batteries that all of our cells rely on, all right? It's a nitrogenous compound like in your DNA, right? Adenine, it's got the 5-carbon sugar, and then the business end of these molecules are these phosphate groups. These are high energy molecules. And these lines, again, remember, are covalent bonds, some double bonds here, but the covalent bond between this phosphate and this oxygen, multiple ones linking them together, and they're super charged up, all right? High energy, ready to do something, all right? Some sort of work. That's what we're hoping for. Okay, so here's glycolysis. Again, universal pathway, all right? We start off and we have super simplified this. We're only showing you some carbons here. Remember, there's hydrogens and oxygens poking off of these molecules as well. We're not concerned about that stuff. We just want to see what happens. Really, what's happening to these carbons as we move through here? We know it's going to go off to CO2, right? How do we get to that point, right? So up at the top is our glucose molecule. And just as we started this talking about the different reactions, right? Catabolic versus anabolic. This is a catabolic reaction, right? And it takes a little bit of energy to get this stuff happening, right? We do say that these catabolic reactions are spontaneous, but you got to have something that kind of initiates it, right? So I like to think of it as, you know, it takes energy to make energy. You don't just get it by itself, right? And in order to do that, this is what initiates it, right? We phosphorylate this sugar molecule. We energize it, right? There's no enzyme that's making this happen, that's speeding all of this up. Well, disregard that. It's all whatever. All right. 2 phosphates are going to be donated from 2 ATP molecules. When the ATP donates, it becomes ADP, adenosine diphosphate, and the 2 in front of it is just saying we got 2 of these molecules participate. All right. So what happens initially is these phosphates get stuck to both sides of this ring molecule. So now you got a charged up bit on one side of it, a charged up bit on the other, and this big 6-carbon molecule is now supercharged. I like to think of it like you're stocking feet on the carpet, getting a bunch of static electricity as you drag your feet across the rug, right? And you shock somebody, all right? That's what we've done with this glucose. We put a supercharge on this sucker. And as the phosphates get put on both ends, it makes the glucose super unstable, and that sucker rips in half, all right? So now this glucose becomes two molecules, two glyceraldehyde 3-phosphates because of where the phosphate is at. And this technically is already a little bit simplified, all right? Because they don't both start like this, right? One of them's got it on the number 3 and the other is on the other side around, and we manipulate that molecule to give us this, right? We're not concerned about that, right? We get 2 3-carbon molecules that are phosphorylated, right? So they're still charged up. They still have some energy associated with them, all right? And we're going to manipulate that, all right? So both of these molecules, we're going to go through and we are going to use some electron carriers and we are going to recharge the ATP molecules that we used up above, all right? And we're going to create pyruvates, 2 of them, 2 3-carbon molecules. We started with 1 6-carbon molecule. Now we get 2 3-carbon molecules. The carbon is all still in the mix, right? And the phosphates are off of it. So we've generated some hydrogen carriers. This NAD is a special electron carrier that's going to transport the hydrogens and some charged up electrons into other steps in this reaction, right? So a lot of the time when we're thinking about breaking down molecules, it's generating boatloads of these. And we're going to use those hydrogens to do some work a little later on, right? We recharge our ATP molecules, but if you look here, we have charged up more, all right? Up above, it's taken us 2 ATPs, all right? And down below, we've just generated 4 ATPs, all right? So glycolysis, which is what we're showing right here, takes a 6-carbon molecule, uses 2 ATPs, and when it's all said and done, we get 2 NADHs carrying these hydrogens, and we get 4 ATPs, all right? And if you do the math, it took 2, you got 4 overall, we made 2 ATPs, all right? A minuscule amount of energy, all right? And if we're talking fermenters, that's what they're getting out of it, all right? Very low amounts of energy, but it's enough to keep alive, all right? We can survive this way, all right? Not super great, all right? So getting the pyruvates is the end goal for glycolysis. And there are numerous reactions to manipulate these molecules to both make them look the same, right? We don't care about that. 6-carbon to 2 3-carbon molecules make a little ATP. At that point, and we're still talking aerobic respiration, right? At that point, we now want to take those pyruvates and we want to manipulate them. We want to use them, all right? So the citric acid cycle or the Krebs cycle remains the same, right? Citric acid is the first molecule as we move into the cycle that we're going to generate, and that's why it's called that. Krebs was a researcher that ultimately elucidated this process, all right? So we'll look at that, we'll see what's happening, and again, we're manipulating these molecules, right? So this stuff is going to be happening in the mitochondria, right? So we're talking eukaryotic cells. That's what we're doing, right? There are lots of participants in this. And again, we don't want to get bogged down with all of the different enzymatic reactions. I'm showing you a couple of things up here, but bear in mind, there's much more going on, right? And the details of that truly, for what we're thinking about, aren't that critical, all right? So the pyruvic acid, before we get into the mitochondria, it gets manipulated a little bit. It interacts with a coenzyme A, and this is an enzyme complex that forms with this pyruvate that helps to manipulate it and transport it in, all right? So as this happens, this enzyme basically pops off one of these carbons, right? This is our first step where we see a CO2 being generated, all right? And remember, at the end of this stuff, there's 6 CO2s that we get, all right? And there's 2 pyruvates moving into this. So 2 CO2s are getting generated at this point, right? So here we lose a carbon, and we harness a hydrogen in this process. That gives us a 2-carbon molecule, which is an acetyl group, and it's linked up to that enzyme. So acetyl-CoA is what we have created, popping off that carbon and sandwiching it into this enzyme. This is what shuttles it into the mitochondria, right? So the coenzyme helps to kind of facilitate it getting into that mitochondria. Now, once we are in the mitochondria, this is happening in the matrix of the mitochondria. So in that inner bit, the bag within the bag, it's in that second bag. That's the matrix, right? As this is happening, there are all sorts of enzymes in the citric acid cycle that grab these molecules and manipulate them, all right? It doesn't matter if we're talking respiration or photosynthesis. There are similar cycles. They are not the same, and they are not just the cycle in reverse, all right? But similar ideas of breaking and building molecules to different types of cycles. And I like to use the analogy of sourdough bread. You can get this starter culture of sourdough, and you can maintain that basically indefinitely, all right? Once you have got your sourdough starter, you pull that out, you allow it to warm up a little bit, you get all your other ingredients, and when you would be adding yeast, if you were making bread, you add the starter culture. And it's got the yeast and other bacteria that make the sour, all right? And you throw that into your dough, and then before you're ready to make whatever you're making, you take a pinch of that stuff back, and you throw it back into your starter culture. So you've got a quart jar that's basically always full because you take a little out, but you put a little back, right? And it's the same with these reactions. They constantly are able to turn as long as we've got an input of specific molecules. And it doesn't matter. Respiration or photosynthesis, these things function like that kind of starter culture, keeping the cycle going, all right? So there are numerous things as we manipulate these molecules, generating carriers, more carbon dioxides, and a little bit of ATP, all right? So, and again, in the mitochondria. As that happens, and at this point, now we've lost a CO2, here's 2 more. Now that all of the carbons are gone, all right? Now what we're left with, carbons and oxygens, I should say, what we're left with are the hydrogens. Those 12 hydrogens have been snagged with NADs or FADs at the first step and in this step, and we've charged up these carriers, right? Giving them boatloads of hydrogens that they bring into this next step, the last step, all right? We call it oxidative phosphorylation. It's also called the electron transport chain, right? We are moving these hydrogens. And it's like active transport. We're going to push them against their concentration gradients. We're going to shove them into that inner membrane, the bag within the bag, in between the 2 bags, all right? That's where it's going, all right? So we'll see this process. And it is busy, all right, as we move through here. Even though I told you we're trying to simplify it, it's still pretty busy, all right? So this is the matrix, right? The bag and the bag inside of it. Inside of that second bag is where the citric acid cycle is happening, right? Break all of that down. We generate all of those hydrogen carriers, and those hydrogen carriers interact. All of these blue guys are different enzymes, different proteins studded on this inner membrane, right? And the hydrogens, the NADHs, interact with these enzymes and they dump their hydrogens. It's basically like a little pump, all right? And the energy that they've stored up forces them through into this inner membrane space. So now we start pumping this area full of hydrogens, right? And you get a bunch of positively charged hydrogen ions. You're supercharged, highly energized, all right? There's a lot of potential energy. It's like water behind a dam, all right? And the more hydrogens we shove there, the more potential energy we have, all right? So we've supercharged this area in this space, right? Full of hydrogens. And then this guy, which would be over here someplace, we're not showing it right there. This guy is a step down the road, all right? So we get a bunch of hydrogens in there. Now we're supercharged. And this enzyme is called ATP synthase. It's going to synthesize or recharge the ATP molecules, all right? As you get this huge potential energy in this intermembrane space, the hydrogens move through here by diffusion, down from high concentration to low concentration. It takes no energy now. They move through this this protein, and that energy, as they are trickling through that, is enough to put a phosphate onto an ADP, and it recharges it, all right? And it's this point where you get the bulk payoff. A huge amount of ATP is generated in this process, and the others just giving you little bits, right? So aerobic respiration gives you a huge amount of energy, right? And the fermentation that we started with only gives you a little bit, all right? There's definitely advantages to aerobic respiration, right? With that said, there are other mechanisms that we call anaerobic respiration, not fermentation, all right? And some organisms, some bacteria are anaerobes, but they will respire. They use something instead of oxygen as a final acceptor to make a bunch of water molecules. They'll use something else, like maybe sulfur, and they dump the hydrogens on the sulfur to make H2S, hydrogen sulfide gas, right? Rotten egg smell, the stink bombs, right? That's what that stuff is, right? So there are fermentation, there are respiration, and truly, when we think respiration, it can be aerobic with oxygen, or it can be anaerobic with something else, and you're still getting a huge payoff, all right? Lots of energy, all right? Fermentation only gives you a little bit of energy, all right? So we'll look a little more about fermentation, all right? And as I said with this, think of like our own body, our muscles, as we go through these processes. Technically, we would like to fully respire. We would like to have ample amounts of oxygen, and we would like to get as much energy as we can. But if cells start to become starved of oxygen, they still want to perform, and in turn, they switch over to fermentation. Not enough oxygen, we ferment. And in the process of doing that, we start generating lots of lactic acid or lactate. That, if we're thinking athletic stuff, it accumulates in your muscles and it lends to fatigue. As you're thinking like a marathon runner, toward the very tail end, almost at the finish line, there's so much lactic acid that try as they might, they can't even move and they collapse because it's accumulated so much. I say, why do that to yourself? You should just sit on the couch and have a frothy beverage. Things would be much better, right? But that's not the case, right? So we go through glycolysis, generate the pyruvate, and then we ferment. And ultimately using the pyruvates as these acceptors. So we get the hydrogens and then we donate them, generating that lactate. It's still the lactates, 3 carbon molecules. We haven't done anything else with them, all right? And there's a lot of energy involved. So later, this stuff ultimately goes to the liver, and the liver will aerobically respire. It will manipulate these molecules, but it takes time, right? It doesn't happen immediately. That's why your muscles stay sore, right? Until you can manipulate and metabolize that. With alcohol fermentation, it's a couple of steps, not just straight pyruvate to lactate, right? The pyruvic acid does get metabolized the littlest bit. We do release a single carbon dioxide off of each pyruvate. That generates a 2-carbon molecule called an acetaldehyde, right? Hangover maker, you better believe it, all right? The acetaldehyde, this molecule, gets manipulated and generates the ethanol, right? And if we are manipulating or metabolizing the ethanol, as we start to break that down, we generate aldehydes in a process of metabolizing. And that can lend to issues, right? So the ethanol is a 2-carbon molecule. We've lost a little bit, right? And if we're thinking frothy beverages, that's where that comes from. The CO2 that's being released adds the carbonation, right? Or just in the process, in this case, making wine, it initially starts off with lots of gas bubbles that are being released. If you take that product and then add a little sugar or other stuff at the end and bottle it up, that's when you get some sparkling wine. Or if you are in France, you could call it champagne. Can't call it that in America. It's sparkling wine, but it's still champagne, right? It's the same stuff, it's just carbonated. If you were making beer, you would have much lower alcohol content in the first place. And as you are going to bottle it, it's the same idea. Add a little malt or sugar to that mix, cap it up, and it will carbonate. And you'll have a frothy beverage. So here I mentioned the anaerobic stuff. So anaerobic cellular respiration utilizing other stuff. So the hydrogen, and we see here the hydrogen sulfide as the example. So something else, right? And we think about these processes as more kind of ancestral, right? Before oxygen was abundant, anaerobic process was the main way that we were going about harnessing this energy to begin with. Similar idea with electron transport chain and all of that. It's just the molecules in that last bit are different than oxygen as far as what we're going to be using as a final acceptor, but the end goal is basically the same, right? So we think about with this whole process, and oftentimes when we describe this stuff, we think about glucose, we think about sugars, right? But I'm pretty sure not a single person in here says sugar is the only thing you eat, right? Even if you're a little kid and you love sugar, that's not going to be the only thing they're eating, right? It better not be. There's problems, right? So this process, as we are manipulating these things, we think of carbs as some of the main stuff. And as we move through here, that's what we've really focused on to begin with. But there's other stuff that we can manipulate and metabolize as well. Some amino acids and glycerols, which are part of these fatty acids that hold them together, the lipids, right? The glycerol bit of it can all be brought into glycolysis, and we can manipulate those carbons that way. Fatty acids and some amino acids can move into the pyruvate oxidation step. So right before we're pulling it into the mitochondria, right? So we can manipulate some of those molecules that way. And then some other amino acids might shuttle into the citric acid cycle. In all of these circumstances, we're thinking catabolic reactions, we're breaking stuff down. So sugars, proteins, and lipids, all on this list. It's not just eating sugar, all right? It all can cycle in at some point. And just as I kind of was describing, we're seeing stuff as the breakdown, but we could flip it around and think about building these molecules as well, all right? Putting it into reverse, all right? Generating other stuff. Oh, I put this in here too. I didn't realize it was at the end of this. It's on the end of the other one because I scrolled through that a minute ago. So this, I remember as an undergrad rolling into biochemistry, and there was a poster of this on the wall, and I was just like, holy cow, should I just turn around and leave? If I have to know all of this, I think I don't want to. we definitely went over these things, but it was not something that was committed to memory. And if it was purged away with alcohol. All right. The idea with this is to just show you all of the stuff, every one of these, well, it's different stuff that we're looking at where we color coded, but all of this stuff where you're seeing all of these words, those are different enzymes, different reactions, and we're either building or breaking stuff down. Coming right down the center of this is what we just talked about. Glycolysis, citric acid cycle, and electron transport chain. All of that stuff is happening right here. And then the other stuff, the outside, is how we start building different amino acids, making proteins, building lipids, all of these different things, the anabolic end of it, right? We're not going to get bogged down with all of that stuff. All right, questions or concerns? I see no questions on here. Let me go to this. Because we still have about a half hour. Okay, that's perfect. So really the idea with this chapter is, where do we get the sugars? That's the whole idea. We went through and thought about how we break these things down. Now we want to think about how does it get made in the first place. And it's not the exact same as the opposite reaction, but it kind of looks that way, all right? It's a little bit different, right? So there are all sorts of ideas, right, with this. And we ultimately, everything is relying on organisms that are able to photosynthesize, right? Whether it be to tap into oxygens or to the sugars that they're producing. And oftentimes it's both, right? So we think about stuff like plants, for the most part, when I think about stuff like photosynthesis, that's what I think about. But algae, as we see here, or some cyanobacteria, all of these guys are photoautotrophs, using the energy from the sun. And an autotroph means a self-feeder, all right? So using the sun to feed itself, photoautotroph, all right? And in the process of doing this, they generate sugars and they generate good quantities of oxygen that we tap into for cellular respiration, right? So we utilize this. We need all of this stuff, right? And it moves through a food chain, right? Which we will talk a little later and describe maybe more web-like than chain-like when we think about energy moving through. So here we can see a grocery store, and photosynthesis ultimately important as we look at numerous products in here. Most all of these with, some sort of corn derivative as a product in there in the first place. Even as you're looking at it, you're like, well, this isn't the produce section, right? There's all sorts of stuff, right? Lots of carbohydrates and probably a boatload of high fructose corn syrup you could find in all of this stuff, all right? If you start looking at labels, right? So photosynthesis is broken into two parts, the photo part and the synthesis. The harnessing of the energy from the sun and the producing sugars in the synthesis bit, all right? There are numerous things that pour into this process. The energy from the sun is what's going to get molecules manipulated and excited. We're going to be pulling in carbon dioxide from the atmosphere. We're pulling in water molecules through the roots. We manipulate these guys to build sugars, and we manipulate ultimately the waters to release this oxygen. And we'll go through this, kind of like the cellular respiration, some extra details as we move through here, all right? So this looks kind of like the opposite of cellular respiration, and it should, right? For photosynthesis, it takes carbon dioxides, it takes waters, and we make sugar and oxygen, right? And in cellular respiration, we saw these as the reactants, and this is the product, right? It looks the same, but in reverse, right? And for what we want to think about in here, it's really kind of like that, all right? It's kind of just like the reverse reaction. Deep down, it's not exactly like that, right? And ultimately, in these reactions, there's more water, and there's also water over here, all right, that is participating in these reactions, and it's also being released as water. So it's a little different, all right? Plants ultimately, cooking out there in the heat of the day, lose some moisture through evaporation. So it's got to kind of fit into this equation as well. And with that said, we'll see here in a second, not all plants photosynthesize exactly the same. Desert plants photosynthesize a little bit differently to minimize the amount of water that they lose. And other plants, well, they technically don't really care as much. And they will spew out water, even if it's cooking hot. And that makes them maybe not so good in some of these arid environments. So not all cells of a leaf are able to carry out photosynthesis. There are different bits if we were to look at it across section, different areas. We showed a little bit about a plant cell. We showed a little bit about chloroplasts before and a cross section of these guys. There's more details as we look at this leaf and see what's happening here. All right, so the top portion of the leaf, the upper epidermis, like your skin, all right? As we move into the inner bit, this is the mesophyll layer, and we've got a handful of different types of cells. Down on the bottom, the underside of the leaf, is the lower epidermis. And there's a little bit of importance down here. There are some openings, little pores. We call them stomata. It's the mouth of the plant, or mouths, because it's full of them, right? On the underside of these leaves, these pores allow for gas exchange. It allows carbon dioxide to come into the plant, and it allows oxygen gas to escape from the plant, all right, through these little mouths, all right? And as I said, not all plants photosynthesize the same. It has to do with what you do with your mouth, all right? Some plants keep them open in the heat of the day. In that case, you're going to lose a boatload of water, right? Desert plants shut their mouths when it's cooking hot. And in this case, they dramatically reduce the amount of water that they lose. A giant cactus can live in the cooking hot desert because it keeps its mouth shut when it's daytime, right? That makes photosynthesis have to happen a little bit differently, right? But it happens, right? So we're not going to worry about that stuff at this point, all right? As we move in here, it's these regions that hold the chloroplasts, and all of these little green guys in here, in this mesophyll layer, where the photosynthesis reactions are occurring, right? So it depends, right? And I mentioned the autotrophs, self-feeders, right? Capturing that energy. So we're not going to get bogged down in this, but the energy moves in waves, right? Photons, if we're thinking our solar energy, little packets of energy, right? And a wavelength is from the top of one hump to the other, right? One trough in between, that's a wavelength, and the crest is the top of that hump. The closer these wavelengths are, the higher the energy that specific stuff has, and the farther apart they are, the lower energy it has, right? So the visible spectrum that we can see right here is just a very small fraction of the electromagnetic spectrum, so the full amount of energy, all right? So over here you can see very long wavelengths, right? Very low energy radio waves, right? Infrared, and then we get to the visible spectrum, right? After the visible is higher energy, ultraviolet rays, sunburn makers, skin cancer givers come at this point. And then we move to even higher, X-rays, and then finally to the gamma rays, super high energy, all right? Okay, and it's this bit, the visible spectrum, that we think about being able to energize these chlorophyll pigments, these molecules inside of the plants, to excite them and get them to do something, all right? Get them to do some work. That's the hope. That's the idea, all right? So there's all sorts of potentials when we think about this stuff, all right, as they're pulling in light. And, you know, as we look at this forest, there's definitely issues where parts of the tree are not fully able to photosynthesize. Some of it gets shaded. Some of the, you know, the forest floor, dependent upon where you're at, has lots of stuff to grow. Others where you've got a massive canopy, thinking maybe tropical rainforest, not much on the forest floor because not enough energy from the sun's able to make it down to the forest floor in the first place, right? So it truly kind of depends on those circumstances. And as that energy from the sun excites these molecules, there's handfuls of reactions that are occurring, all right? So this, again, inside of the plant cell, all of these little chlorophyll molecules, the energy from the sun exciting these molecules, these chlorophyll pigments. And it's kind of back to what I was describing before with your socks, dragging them across the carpet, getting a bunch of static electricity. You're super charged up and you can shock something, right? It's a similar idea. This energy from the sun supercharges these chlorophyll pigments. And what they're going to shock is a water. They take that water molecule and they drive that energy into the water molecule and they split it apart, generating oxygen gas and some hydrogens. And we showed in the last chapter, those hydrogens were important to move them around and do stuff, right? Oxygen's important here because we want to breathe that stuff, right? So we know that the water gets split and it gets split in the photo portion. The energy from the sun splits it apart, right? There were researchers trying to figure out in a plant, where's the oxygen come from? We know plants need carbon dioxide. We know they need water. Both of those molecules have oxygens associated with them. So they did a couple of experiments and grew plants in a greenhouse in a special chamber where you could control what was happening. And one of the plants was watered with oxygens that were radioactive, and the other was supplied the carbon dioxide with its oxygens being radioactive. They allow the plant to grow, then they sample the plant and see which plant has sugars that are radioactive, all right? And in turn, those that were, or not sugars, has oxygen, has the gas released that's radioactive. So the plant that was watered with radioactive water had oxygen released that was radioactive. And the plant that was given radioactive carbon dioxide, it had sugars that were radioactive because those oxygens in the CO2 get incorporated into the sugar molecules, right? So water gets split and that generates this whole energy needed to start making sugars, all right? So this again is in the chloroplast molecule. This is similar to like the mitochondria, but there's different players, right? Light energy exciting these chlorophyll pigments. There's a couple of different pigments. We're not going to get bogged down with this stuff. Different amounts of energy, wavelengths that they can be harnessing. It's slightly different. They're labeled 2 and 1. It was basically order of discovery, but we know the second one happens first. These are things that I'm like, this is what makes people hate biology. Just put this shit in order. Right? Wouldn't that be easier to understand? I think it would. So 2 happens first, then we go to 1. Right? As this occurs, we excite that energy, we generate hydrogens, and we're going to move electrons through a handful of proteins, continuing to excite some more of them, right? And then finally generating, like our NADH, this has just got a phosphate associated with it, but it's the same idea, all right? So we generate this carrier. This is going to help build sugars. And we also, as we're pumping hydrogens in here, generate ATPs, right? These ATPs are going to help build sugars as well, all right? So this is inside of that, the chloroplast, the thialkaloid membrane, those little discs. And then this is the fluid that they're in, the stroma, right? So the carriers and the ATP is there, CO2 is going to come in, and we're going to link them all together, right? So this is, again, that chloroplast, all the little thialkaloids, and then the stroma is for these guys are sandwich in. So this is all the stroma. For the sugars will be generated, right? And that is this, right? The Calvin cycle, it's kind of like the citric acid cycle, right? Where we're going to turn this cycle, pulling in CO2s and generating glucoses instead of the citric acid where we're pumping in pyruvates and releasing CO2s, right? Same but different, right? So the carriers and the ATP pour into this along with the CO2s and we generate sugar, right? That's the idea as we move through here, right? Numerous steps, numerous enzymes that come in and link this together. This protein up here, Rubisco, is what's snagging the carbon dioxide. It is a molecule that is seen in most of the plants. There are a few that, like I said, that photosynthesize a little differently. There's different protein, right? But Rubisco is the most dominant protein in the world because the majority of plants photosynthesize by what we're describing in the first place, right? So it snags the carbon. This is carbon fixation as it grabs the CO2 molecules. Some of the ATP that we've harnessed from the sun gets used to manipulate these molecules, rearranging them. We're turning Calvin 6 times, 6 CO2s coming in to pump out a glucose molecule, right? As we are turning, we keep, just like I said, the sourdough culture, keep it churning, creating these 5 carbon molecules, ribulose bisphosphate, that in turn keeps churning as Rubisco dumps the CO2 onto the ribulose bisphosphate, generating a 6 carbon molecule that we in turn start pumping off, right? So it doesn't just happen in one cycle, it's 6 cycles, 6 CO2s need to be incorporated to build this because we constantly keep regenerating, right? It doesn't happen in just one shot or you'd run out of starter culture, right? So, oh man, we are booking through this, which is exactly what I wanted to have happen, all right? I didn't want to say it at the start because I didn't want you to get like just expect it. But my hope is we will wrap all of this up and then Thursday you guys will just not be here. As it turns out, I have another class that I technically need to be at. And I was like, we'll just start them a little bit late. But I really didn't want to do that. And I think there's just a few left. So we'll get done with this. You will have Thursday off. When you could take the test, right, as class time should be happening. Will you? I bet not. But you could, all right? Will you take more days to study? Probably, all right? I think you ought to just take it. All right. So the cactus was my example as some that do things a little bit different, all right? Keeping their stomata shut, all right, when it's super hot. So at night, when temperatures are a little more manageable, when the potential to lose a bunch of moisture is not as much, they open up their stomata. They pull in the CO2. Then as the sun starts to come up, they shut their mouths, right? And now we start going through the Calvin cycle. And we slowly release those CO2s and generate the sugar molecules. Cactuses and other desert plants succulents, things like that, take a lot more time to grow because of this process of photosynthesizing a little differently, all right? The grass in your lawn, most of it is a photosynthesizer like we just described. And in the heat of the summer, it tends to get kind of unhappy, tends to burn up a little bit because it's losing a lot of moisture. Quack grass or crabgrass is a different photosynthesizer, different than the cactus as well. And it shuts its mouth partially, right? Losing a little bit of water, but not a boatload, all right? And it still grows really well. Corn and tomatoes are also photosynthesizers like that. We call them C4 photosynthesizers. They make a 4-carbon instead of 3-carbon molecule. Doesn't matter, right? And this, with the cactus, is a CAM for crasulinic acid metabolism. You don't need to know that, all right? It's just a different type of mechanism. So this is showing with our little prokaryote how these guys initiated the ability to photosynthesize in the first place, forming this precursor thiopolate-like substance, but not anything we need to get bogged down with. So this image, and I think it's not, well, really, you've got that chemical pathway in a second, but this puts chapter 4 and chapter 5 together in one slide. We think of photosynthesis producing the oxygen and the sugars. We think of cellular respiration, tapping into those sugars, releasing carbon dioxide and water, and ultimately both are reliant upon one another, right? I think deep down we would like to think that's a straight, 50-50, we're both equally reliant, but I think really we're more reliant on the plants than they are on us, all right? I think they could do just fine without us, all right? But we can't do just fine without them, right? That biochemical pathway, I thought that was on this chapter too. I lied. Okay, any comments or concerns? Let me stop my share. Okay, anybody tuning in from home? A question here, yes, sir. Do I have that posted on the syllabus? I think I do. I think I want to say Monday. Are you saying Monday?
Yeah,
Monday at 5 p.m. Because I've learned from my mistakes and I don't close tests at 11:59 p.m. I don't need the nonsense. 5 o'clock. So the test will open Thursday morning. There is no class Thursday. You can take the test during class time, or you have until Monday at 5 o'clock. You need to get on Moodle. It is open book, open note, not open neighbor, and not open Google.
Sorry, you
Right?
said it was open book.
Open book, open note. And it doesn't matter if you've got a hard copy or an ebook. You can use the PowerPoints. You can open those suckers up.
Notes too.
Notes, PowerPoints, book, all of that is fair game. Not neighbor. I don't want to see you Googling stuff. I will see what you're doing. Yes. What? Yeah, it'll be on Proctor Free, so it'll be right. You go on to Moodle, you'll see it'll be loaded right into that. So you click on your Proctor Free, and it will have the first exam there. You'll be good to go. Yes, it will flag you as cheating, but I know you're not. It's just fine. It knows what you're doing.
Well, so we're allowed to go between like Google. The exam.
Yeah, yeah. It should be smooth sailing.
And if we have like notes on Google Docs, we should be good too.
Yep, it will know. It knows what's opening. It sees all of
I
that.
don't know if I'm like missing anything, but like I go on there and is this like the first test that we're doing?
Yes.
Okay.
Yep.
Just double checking because I had not seen.